to the other room because in the remaining part, we would like you to start to do things hands-on. So it will be with Ahmed and Tim. But before we move there, a few more things, a few more background issues I would like to discuss, and then we will do that. Now, one thing we have considered is the following. You know, in the PDB, originally when people were trying to resolve structures, the role of computational biologists was to let them know which particular sequence is a good candidate to obtain a new fold, a new structure. You may know that. Things changed since then. And what happened actually, in principle, is we have in the PDB lots of structures for a number of very well-studied proteins, lots of structures in different forms. In the presence, for example, for many drug targets, we have many structures in the presence of different drugs, or in the presence of different ligands in complexes, etc. And we actually found it extremely useful to have those ensembles of structures. They're useful because, why? They give us information on the conformational variability, you know. This is the same protein, let's say p38 kinase, or ubiquitin, which Klaus described. And now you have a multitude of conformations that are accessible to it, depending on the protein it binds, on its substrate. And then we wanted to see whether these ensembles of structures, are they any relevant to what the alternative conformations that we're generating? Because these ensembles of structures give us information on what the protein can do or is doing in the presence of different substrates. And we wanted to see whether they correlate with the modes that we are predicting. Okay? Now, we have ensembles for our proteins in the presence of different substrates. Or we can have some structural changes induced by ligand binding. So for p38 MAP kinase, which is an important drug target in the presence of different inhibitors, you have all these different types of structures available. Or then we can move even to bigger systems. That's something I mentioned earlier, a molecular chaperonin, GROIAD, bacterial, which is composed of, overall, there are 7 subunits in this lower ring, 7 in the upper ring, and then there is a cap. So overall, 21 subunits. But we can even have structures composed of hundreds of subunits, like viral capsids or the ribosome. And we have now ensembles of structures that refer to different conformations that are being visited along the allosteric cycle, along their functional mechanisms. Okay. Now, if we focus on that one, on the GROIAD, for example, it is possible to view more closely what is happening, and this is what I was mentioning when I was answering you. At the interface, for example, we can focus what's happening at the interface, and this movie, this simulation, is just generated by using as steps the collective motions. So think of in MD simulations, you know, each step you evaluate each pair of atoms and you use one atom at a time. Now think of a much faster methodology where you move simultaneously all atoms along the modes that are intrinsically favored. So if you have such a multitude of modes and you use them in a cyclical way here, you can observe what's happening at the interface. What's most important is that you can observe some changes, redistributions of interactions. Here is an example from, again, for the same interface. What's happening in the first mode, in the slowest mode, is this, you have a subunit here, another subunit here, and a third subunit. And you have at the interface many salt bridges, okay? So originally, this salt bridge, now it's going to be dissociated, and there's a new salt bridge that's being formed. Okay? So that is critically important. if you mutate one of the particular salt bridges, that was the one that was originally formed in the wild type, if you mutate the corresponding glutamate residue here, then it is very likely to rearrange itself to find this new partner and now be locked in this alternative conformation. Because it's a soft mode, it can do that easily, and in the presence of this mutation, it's going to do it, and it will be locked in that conformation. As you can see here, you have these single subunits in a staggered position with respect to the other lower subunits. In the mutant, the position changed. Okay, that's another interaction. So what's so important about it, you may ask? But the thing is, this salt bridge, original salt bridge, was very important in transmitting signals from the ATP binding site here all the way to the cap, and enable the continuation of the allosteric cycle. Now, in the absence of this particular interaction, the proper salt bridge network, experimentally, what's being observed is that the structure is locked in this dysfunctional conformation. Okay? So it's very important to see what the ability of the molecule is, and why certain mutations are deleterious while others aren't. Okay? Yes?
So would those residue be identified as the peak of the noble mode correlation?
No, actually, they are, the molecule is moving. This is like the interface. They're not the peak. On the contrary, these are the regions where there is a small motion, and the motion, as you go away from that interface, it gets larger. So the peaks will be high mobility regions. We're usually more interested, the critical sites are the minima, not the maxima. The maxima would be the recognition sites, but the critical sites are things you don't want to interfere with.
In one of the plots that you showed before, you just put an analysis and looking at the different sizes of the modes.
Yes.
You identified one as, let's say, hinges, for example.
Exactly.
How do you know that they are hinges? Is it the particular value for the modes that identifies them as hinge?
It is a hinge. We say this is a hinge because it is the size of the motion there is 0. So it is.
Oh, okay. So the 0 ones are.
The 0 is a hinge. So this is like, I was showing my elbow, which is not moving. Of course, if it were not attached here, I would have both sides moving. So this is another hinge, for example. Yeah. The hinge is not moving. So in the structure, suppose you have this type of movement, and in HIV reverse transcriptase, there is the palm and finger and thumb, which I'm going to show. The palm region is the hinge. And that's where the reverse transcriptase inhibitors bind to block this movement, actually.
And you remove the trivial motions, right? That's why, because the hinge I can translate it. Or
Absolutely, absolutely. Yes, yes, yes. And sometimes, there are subtle details. For example, in the isolated molecule, you have this type of hinge motion. But suppose now it is embedded in a membrane. This part is not going to move then. And this part is going to move even more, Sometimes stabilization of one part may induce even further movement in the other side. So it's concepts like that. Okay, so this is, again, my favorite molecular chaperone. And it's this trans and cis forms. I'm sorry, this is the cis ring and trans ring, and this is the relaxed form and trans forms. There's a transition between them. And I'm focusing only on the individual subunits, and you can see the alternative conformations that they have. This is the top view again, and these are the alternative conformations of the adjoining subunits in the relaxed and trans forms. And if I compare, I can evaluate for this particular transition of this subunit how much each individual residue has been displaced, okay? I can evaluate what's called a deformation vector. This is pure experimental data. Now, this deformation vector, I'm going to compare with what? With our fluctuation vectors induced by individual modes. So here are a series of individual modes. And I'm comparing this experimentally observed deformation with my series of individual modes. Normally, the modes form a complete space. They form a basis set, you know, for. So if you take all the modes, if you sum them up, you can always reproduce any motion you like. But we would like to see whether this motion is enabled by a soft mode, right? And that is, and we see, look at the correlation cosine between all of them. And look at the correlation cosine with the first mode in this case. So whatever the change that is taking place in this particular case, we're not so lucky always, but this is a good example, where the change in the structure is essentially nothing else than a movement of the molecule along the softest mode accessible. So that's the value of the, now if we know what is the softest mode, we could predict that it is likely to do this type of, and it's very important to understand what are the conformational changes, especially for drug binding. These are moving targets. And the other, this is the cumulative overlap that we have. So this is the correlation cosine. You take the square, and then you add up the squares of all correlation cosines as a function of mode index. And eventually, it's going to go to 1. Okay? But it's, you already accomplish 70% of the cumulative overlaps, correlation square, or more than 8% correlation cosine by a single mode. Yeah?
Can you, in particular, give it two structures and say, I want to move from this structure to that structure? What are the modes of getting there?
Absolutely, absolutely. Now you know the deformation vector, okay? You know this vector, and you know your eigenvectors, individual modes. You can take one by one, you know, see the correlations. And I know that I need to move a lot along this mode, and perhaps get some help from mode 2. And then I, you know, or, but I'm not, you know, we're not always, we don't want to go all the way. To understand that this is the basic mechanism, the most important change is already, and after a while, things also start to hit the resolution of the structures, you know. So this is another example I mentioned, the HIV reverse transcriptase, from a study I did almost 15 years ago, where we have multiple structures for the same HIV reverse transcriptase. And here is, for example, this is what's called the thumb. This is like a hand, thumb. These are the fingers, so I don't know, like that perhaps. Fingers, and this is the RNase H, and here is the yellow is the palm, and the little thing that you see here is the drug. So the drug binds the palm, as I mentioned earlier. And this is the RNA binding. So it closes down on it, so it has this type of global mobility to do its function. Now, if what we did actually was to take the ensembles of structures that have been resolved, let's say for HIV reverse transcriptase. Now we superimpose them. We have some DNA RNA bound, apoforms, or inhibitor bound. So we have a cloud of, this is our ensemble of structures, okay? Now from this ensemble of structures, for every single residue, I know what its mean position is, and I know how it deviates from this mean position. And I know how its deviation correlates with the deviation of another residue. So I can construct from this ensemble the covariance matrix. Are you with me? Now this covariance matrix, remember the covariance and the Kirchhoff matrix, they are the inverse of each other. So you can either eigenvalue decompose the covariance or the Kirchhoff. But now we want to see from this covariance, purely based on ensembles of structures, what are the principal components, and how those principal components now relate to our modes, okay? So the principal component in this case is this motion, which you see with these arrows. So we just did that, constructed the entire covariance matrix. This is only the IJ 3x3 block. We have a 3n by 3n matrix. We did the eigenvalue principal component analysis, identified the principal components and the corresponding eigenvalues. And now we just take the first, which accounts actually for 47% already of the entire variation. And this is the movement, the thumb and fingers moving with respect to each other. Okay? Now, this type of analysis, we've been, actually this is from a study of Ahmed, we have been applying to a series of proteins. And here you can see the pairs of arrows actually correspond. One, I don't remember which color is which. One is the experimental principal mode. The other is the ANM, an isotropic network model mode, okay? So we are doing essentially, okay, we know now which color is which. The PCA of the experimental structures are in violet, and the ANM modes are in green, and you can see how they agree in general. This is from this study, this PNA study that I have already mentioned of Ahmed. So if you come back to our reverse transcriptase, so these are the alternative conformations. And what the ANM predicts, essentially, is the same type of, so this is the structure. And this is the mode 1 that we are observing, or mode 2, I don't remember now. So, and this gives us the correlation. In this case, between, this is ANM mode 2 and PC1, we have an almost perfect correlation. And this is the basic concept. You know, if there is a single thing you need to remember from my lecture is that soft modes are very important to enable functional movements. Okay. And this is just a, you know, a bridge to tomorrow's lecture, perhaps. This type of movements, so these are the alternative conformations of a protein in the presence of two different ligands in red and blue. Depending on this, on the ligand that the protein binds, the inhibitor, you can have different conformations. And this type of conformational flexibility plus sequence variability will be effective in selecting the substrate. And tomorrow we will talk about how this, we can explore the dragability of proteins and how also we take advantage of sequence variations to understand better what's happening. With that, next you will go to the next room to just familiarize with Prodi more. And essentially, there is an overview. One good application, which would be to give a sequence, a certain sequence, and you extract from the PDB all the sequences that are identical within a certain identity threshold. Now you can take the same sequence or just members of a given family, and now you have an ensemble of structures which you can analyze either by our PCA method, or you can just take one representative member and see what it's doing. You can compare experiments and theory, you can identify, you can generate alternative conformers, and then analyze them further, etc. So I will, without further ado, I invite all of you to move to our next room.